All right. So if there are any more questions, please feel free to drop them in the chat. Otherwise, I would like to introduce our next speaker, who is Gustavo Palacios, who will be discussing advanced genomic sequencing for human-to-human transmission analysis of Poxvirus. Welcome.
Hello. Can you see my slides?
I can see your screen.
Let me try again.
There we go.
Okay. So thanks for the invitation to present in this meeting. That was a very nice presentation so far. So obviously you are aware. Obviously we are reunited because of the situation that have been. It's in the process. It seems that it's in the process of limiting the degree of urgency, but it has been highlighted what are the uniqueness on the outbreak, on the monkeypox outbreak that we are living, as it has been related to a unique pattern of transmission, mostly among members of the male-to-male sexual men that have sex with men community. So that uniqueness in transmission essentially has it highlights the reasons why it's very, very important now that we can have better tools to track human-to-human transmission and to try to figure out if those patterns of transmissions are different to what we have observed in the past. So these in terms of research gaps identified by WHO, modeling transmission dynamics and sequencing to identify changes in circulating viruses had been highlighted as a huge gap in our knowledge in the current outbreak. So in terms of our background, what we know about monkeypox transmission, and especially for person-to-person transmission. We know that in the 2010-2013 Congo outbreak, we have seen genomic changes associated with the inverted terminal repeats, and a lot of the speakers have talked about those observations. We know that there are areas of low complexity regions that have been tracked to human transmission in general for pox viruses. This has been observed for variola and other pox viruses already. We know that also that the zoonotic spillover that was related with the current outbreak probably occurred in Nigeria in 2017. We know that content duplication is very important for rapid adaptation of pox viruses, and that has been demonstrated very elegantly experimentally in systems for gene duplications and for base duplications in the genome. And that gene redundancy, the loss and the capture are the main drivers of pox virus evolution. And that has been, a lot of the speakers have referred to the papers from Elde, Senkevich, and Kunin and Moss in that idea. So the concept of genomic accordions had been mentioned in all of these talks, and I'm going to refer to it also. So what are the low complexity regions and the variability that we are observing in low complexity regions during the current outbreak? So I'm representing here one of the short tandem repeats in samples from the outbreak. This is the one that we identified as short tandem repeat number 2. Is a D-nucleotide repeat with a very clear AT modification is near one of the inverted terminal repeats, and obviously it's a very challenge for short-read sequencing technologies, which is could generate a lot of errors in the sequencing. Any amplicon-based systems also is going to have huge problems to resolve this particular area. STR8, another area of low complexity repeat that I'm choosing here to display to you. This is a more complex palindromic region, is is in the core region. And it has, again, a similar, reported genomes have huge differences in that particular area. And in a lot of cases, I must say that a lot of these genomes have been probably receive a bias of the way that the reference assembly that has been conducted and what is the genome that was used to use the bioinformatic tools by the users to generate these genomes. There are others, short tandem repeats. This is a little bit more complex. Now it's a codon, it's a 3 base pair repeat that it's a codon, CAT, and it is in one of the coding regions of the genome. So it's involved, it's in an area of the genome that is translated. And finally, we have homopolymers that are single homopolymers retreat, in this case only formed by Ts in the core regions that will have a transcriptomic consequence in the genome of monkeypox. So how do we, I tried to focus in the 12 minutes that I have for this presentation, how we resolve this, and what we posit to use is essentially avoid amplicon sequencing, work with with tools of direct sequencing that do not have biases during amplification. For that, we have experience working with one vendor, but there are multiple of those that produce beta enrichment probes that could be used to enrich for the given areas of the genome. So we have done this for pathogen discovery and for viral characterization, and we use this product in the past to characterize the original monkeypox clade 2B strain from Nigeria in 2017. So the process of generating a targeted enrichment, I probably do not need to go through the details with this audience, but essentially involves the construction of an RNA-seq type of library or double-strand DNA library construction in this case with then an hybridization from baits that will enrich and generate to pull out the materials that are targeted. So the whole process involves fragmentation, core library construction, where we have identification or introduction of adapters and universal primers, and only after that a process of amplification, which essentially means that we can ensure that that if we are seeing changes or mutations, we can refer to the original DNA molecule to validate that is not a product of the amplification of the library amplification. So we have used this in the past for pathogen discovery and for characterization of other viral infections. But for the use of monkeypox, we have an additional restriction. That is what we want to focus, what we want to see are areas that are highly repetitive. So the way to try to enrich for those highly repetitive areas is to make probes, enrich probes, and generate a higher targeting in the flanking regions of your low complexity areas, and essentially use a concept that has been used in human genomics, that is shadow coverage, to extend to ensure that that bait essentially will pull down the material in the short tandem repeats. This is not a new concept. It is what we with genomics use essentially to resolve the areas in eukaryotic genomes to deal with low complexity areas. So then what you end up having is a enriched coverage where you make probes essentially designed in different areas of the genome, but then you produce and you request that you have 100 times more coverage in the areas where you are seeing these low complexity regions. So why it is worth the pain. I mean, what we win by doing this? Well, first, we are trying to measure what is the entropy that we are seeing in these low complexity areas as a measure to compare diversity. And we are doing that in intra-host, so in the same sample, or interhost, that is in multiple different samples. And what you see here is a comparison of what is the type of diversities that you see in SNPs or in low complexity regions. In the left panel, you are seeing that essentially we have a very significant difference in the number of in the level of entropy, both in homopolymers and short tandem repeats. When we start looking at the interhost variation, that's the intersample variation, essentially we find out that there are essentially some areas of the genome that evidently are moving or are encoding for a lot of more variation than what you see in the SNPs. So we have a lot of variability in these areas. Well, so that is what you can say that it is what we expect, because obviously they are low complexity, and that is why we have a higher rate of error. But next is the question of where is these are located these low complexity areas? Are they informative or not? And what we found is that they are not randomly located in the Poxvirus genome. A lot of these short tandem repeats or low complexity areas or homopolymers are located in areas that are known to be immunomodulatory, that are known to be variable, but they are in a lot of them in encoding regions for these genes. So therefore, housekeeping genes are the ones where we are seeing the lower type of variability. However, some of those short tandem beats are located in areas of core genes, and therefore that would be important to follow up in genotype to phenotype experiments that we are pursuing. So what is what we are missing? So here I'm presenting a very quick characterization of the reference genome that was selected for clade2b, as well as one of the best sequenced samples that we found in terms of quality. That is one sample from the USA from Dr. Gigante that was generated with both nanopore sequencing and Illumina sequencing, and we compare what we call our one of the results of our analysis, that is our high quality genome. And what I am trying to highlight is how there are a significant number of differences in size, a very low number of SNPs differences. A lot of those, several of those differences are referring to STRs, to short tandem repeats that are of a complex nature. They are not in homopolymers. And those are located in STR 1, 2, 3, 4, and 5, where most of the variation is being observed. So in conclusions, I believe that the variability that we are observing cannot be attributed to error sequencing. We have essentially generated this high quality genome using both short Illumina reads, paired 250 by 2 and 300 base pair by 2, and Oxford Nanopore or single molecule PacBio data, to ensure that we are highlighting all of the areas with that hybrid approach to assembly. We get to resolve all of the areas with only one contig. We are observing that there is variation not only intersample but also intersample in the more than 150 samples that we have logged for so far. It is necessary to do this with selected techniques for enrichments and library sequencing, and we are also needing to have a special bioinformatic analysis that essentially relies mostly on de novo sequencing and de novo resequencing to ensure that we resolve correctly all of the short tandem repeats. And we realize that the value of this approach will only be clear once that fully standardized methods of analysis are pursued by others and from samples from multiple different origins. And we believe also that a lot of the comparative genomics that we have been discussing in this session essentially cannot it's very difficult to assess how much of the quality of the data that we are using underlying all of these bioinformatic resources is correct or is seeing the nuances of the poxvirus genomes. So with that, I want to give thanks to the members of my team at Mount Sinai and the members of the Emerging Viruses team at the Center National for Microbiology in Madrid, Spain, who had been conducting a lot of the work that I presented. And with that, I am open to any questions.
Thank you, Gustavo. This is exactly the kind of discussion that we were hoping to generate with all these different sequencing methods and pipelines. Unfortunately, we are at time for our next speaker, so I'm going to have to direct questions to the chat. And